And our group studies clonal hematopoiesis of indeterminate potential, which we've done some work related to this and solid tumors, and we've done some work related to this and kidneys. And so hopefully one of you guys will put those things together and look at clonal hematopoiesis and kidney cancers. But to be very clear, we haven't done that. So it's a wide open space for the trainees. I think, when thinking about our genome, like when I was in college and learning about biology, you're taught that like your genome doesn't change. You inherit one copy from mom and one copy from dad. But that's not true, right? And in the cancer genome world, you guys know that that's not true. We're always acquiring mutations. We have telomeres that are growing and shrinking. We have the mitochondrial DNA that's incredibly dynamic, both in terms of copy number as well as mutations. Today I'm going to talk about one of these changing features, which is called clonal hematopoiesis. And so in clonal hematopoiesis, basically you acquire a mutation in your blood stem cell. And these mutations could be loss or gains of entire chromosomes, like loss of Y. They could be, parts of chromosomes gained or lost, or it could be single nucleotide or mutations, these point mutations, in particular driver genes. And all of these different kinds of genetic events can create clonality in your blood stem cells, which then creates clonality in your fully mature blood cells. And so today I want to talk about four things: diagnosis, what is this clonal hematopoiesis of indeterminate potential? Prognosis in terms of solid tumors and in terms of kidney disease. Sort of a very, very, very short take on how a mutant blood cell can be causing problems, and then talk a little bit at the very end about management, about how we think about this clinically. So CHIP is now an official thing. The World Health Organization included it as part of their classification of blood cancers in the most recent 5th edition that just came out. And the way that the WHO agrees with the community that we should call it is based on three sort of key features. One is that there's many different forms of clonality in the blood, and CHIP is not the same as having a clone. It's defined by having a driver mutation in a specific driver gene that causes myeloid blood cancers, like acute myeloid leukemia. It's defined as being sort of an appreciable fraction of your blood, more than 2% varying allele fraction if you sequence peripheral blood. And that's important because, as you might imagine, the sort of deeper you sequence, the smaller and smaller clones you might find. Every big clone has to have been a smaller clone at one point. And so if you sequence all of us deeply enough to, 1,000,000 X, you'll find these mutations. And by and large, these very, 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 very small clones have sort of minimal clinical implications. So again, 2% is somewhat arbitrary, but I think is a useful working definition. And the other key feature is that they don't have some other blood abnormality that might explain why they have this myeloid driver mutation, meaning they don't have myelodysplastic syndrome, they don't have aplastic anemia, they don't have some other reason why they should have this clone. There's a very sort of closely related entity called CCUS, or clonal cytopenia of undetermined significance, which basically means that you have CHIP plus you have an unexplained cytopenia. And for sort of further thinking about what how WHO came up with this stuff, there's a great review article written by a number of people who are on the committee explaining the nuance in far greater detail than even I understand. CHIP was first described in 2012, 2014 by a number of different groups. And the first thing it was associated with was blood cancer. It's a pre-myeloid malignancy. And then sort of subsequently, it was linked to quite a number of other non-malignant states, including cardiovascular disease, some early associations with lung cancer, and now sort of an expanding array of other diseases. I think what a lot of these diseases have in common is inflammation. And one of the things that we've learned is that CHIP mutations, CHIP-derived myeloid cells like monocytes and macrophages are pro-inflammatory. And so if that inflammation is part of disease pathogenesis, it seems to be linked. So how do we find CHIP? In some sense, it's pretty easy and probably more obvious to this audience that thinks about cancer than other groups. Basically, when we sequence blood, we're looking for mutations that are not 50% or 100% of the blood. So this is a screenshot from IGV, Integrated Genome Viewer. And what you're seeing is that there's a 1 base deletion in DNMT3A in 3 out of the 20 or so reads. And so this mutation is almost certainly not inherited from mom or dad, but it is somatic. And we're able to take advantage of the fact that there have been now hundreds of thousands, millions of genomes that have been sequenced for studies completely unrelated to blood cancer or anything else, studies of diabetes and heart disease and Alzheimer's disease. And we can apply sort of a tumor genetics approach to identify somatic mutations in these genomes, even when they're not meant to find somatic mutations. So many of these exomes and genomes are 30x sequencing depth. But because we have hundreds of thousands of them, we're able to very confidently say, which of these mutations is somatic, which ones are not. And so because we can do this at scale in data that already exists, there's a reasonably sized literature about this at this point that all shows sort of similar variations on this theme, that basically CHIP increases with age. The exact prevalence depends on how deeply you sequence it. If you use whole genomes, such as we have in the All of Us research program, it's about 10% of people, once they get into their late 60s. If you use a targeted sequencing assay, or you use data, that is tumor normal sequencing, like the MSK impact data set, then you can find actually quite a bit more CHIP. It's probably closer to 20% once people are in their 60s. But again, in general, any CHIP data set should have this kind of property that should be uncommon when people are young and should be quite common when people are old. The other sort of very interesting and puzzling thing about CHIP is that driver mutations could occur in any one of perhaps 100 myeloid malignancy driver genes, but they don't. That CHIP mutations tend to be quite common in a very small subset of genes. It tends to be very common in these epigenetic regulators like DNMT3A, which is a methyltransferase that puts a methyl group on a CpG, or TET2, which takes that methyl group and converts it to hydroxymethyl, which is the first step in sort of removing it. ASXL1, which is involved in chromatin regulation. I'd say, so those are sort of the majority of CHIP. Then you have more of your classic DNA damage repair genes like P53 or PPM1D. And then, a smaller but still significant group of splicing genes, which we have in common with the kidney cancer world, like SF3B1, SRSF2. And then what's, I think always striking to me is that sort of your classical oncogenes, like KRAS and IDH, are actually, a very, very small fraction of what's driving this pre-cancerous state. That's sort of the landscape of where we find these mutations. At Vanderbilt, we have a great assay for detecting CHIP that, we basically are able to do a targeted set of about 25 of these genes for about $8 a sample. And as a result, if you or colleagues are excited to do this, we're probably already working with those colleagues. We've had more than 100,000 samples sent to us for this assay in the past year from many of the major researchers in this space, and many sort of epidemiology researchers who are interested in getting into it. And we're happy to extend this to you or anyone at your institution might be interested. But those are really sort of on a research basis. How do we diagnose CHIP clinically today? Really, CHIP tends to be an incidental finding from tumor genetic testing. So we might get a report that looks something like this from Tempus, from a liquid biopsy, or from tumor normal sequencing that says, this was, I think, a head and neck tumor or something like that. And they said, we found a mutation, JAK2 mutation at 3% in the sample. And, we know that JAK2 is not a major contributor to this particular kind of tumor. So probably represents clonal hematopoiesis or maybe some other hematologic malignancy. At Vanderbilt, we do a ton of genetic testing, as I'm sure you guys do at your institutions too. And every week we get probably a dozen or so patients who are being tested for a solid tumor who are coming back as having found a somatic mutation. Increasingly, there's interest in expanding this kind of testing beyond cancer. So there's at Vanderbilt, we have a CHIP clinic. I'll talk about it at the very end of the talk. And again, I know they exist at Mayo and at Dana-Farber and at Sloan Kettering also. But increasingly, there's interest in just doing clinical genetic testing for CHIP, and so that's also pretty exciting. So I'm going to talk about why do we care that 20% of people have in their 70s and 80s have these CHIP mutations, and focus really on two things: on solid tumors and on kidney disease. But before I get there, the sort of first things you might expect is that, thinking about cancer, is that people who have this pre-blood cancer probably are at much higher risk of developing developing a myeloid cancer. And that is true pretty much across every study, that people who have CHIP are at much higher risk of developing MDS and AML. And an interesting observation is that the larger the clone, so the larger the fraction of blood that's mutated, the higher that risk of transformation is. And similarly, the larger the clone, the higher risk of dying is. And these are observations that have been made across quite a number of cohorts since at least 2014. And the puzzling thing, again, in the early work, was putting these two things together. Like, why are these people dying? There's not enough acute myeloid leukemia in the world to explain why, we're seeing this twofold increased risk of death in 20% of people. And so some of those early observations led people to think about, perhaps people are dying of solid tumors. Solid tumors are a very common reason for people to die. Perhaps people are dying of heart disease. And indeed, all of those things are true. The other seed I want to plant here that, again, was observed early in the blood cancer, focus analyses, and has been true across quite a number of different studies, is that the risk of malignancy, of death, of whatever your bad outcome is not the same across all of these different genes. So I showed you that about half of people who have CHIP have DNMT3A as their driver mutation. About a quarter have TET2, and then a smaller fraction have each of the rest of these genes. And each of these different genes have different risks. And so, in fact, we probably shouldn't be just calling this, risk of, disease by CHIP with time. And I think we're starting to do this already. We'll probably be calling this TET2 CHIP and DNMT3A CHIP and, IDH mutant CHIP, and thinking about how some of these different specific mutations are conferring risk, rather than calling CHIP as if it was all the same thing. So, I'm cognizant of the fact that I'm talking to the Kidney Academy. So I want to spend a few minutes talking about CHIP and solid tumors. There have been a number of good reviews, but for people who are excited to dig in more, I call your attention to one by a MD PhD student here at Vanderbilt named Sarah Reed, who published this paper, this review article in Clinical Cancer Research last year. If you think about all the different, the ways that a blood cell can affect a tumor, I think that it is sort of obvious to this group that all of our immune cells are derived from blood cells, and these play a really important role in the microenvironment of solid tumors. I think it also invoke the fact that your T cells also play an important role in immune surveillance, and so it's not just in terms of what's happening in terms of response to different therapies, but also in terms of who develops who has a pre-malignancy in solid tumor space that progresses to malignancy. And there have been some early observations about how, just like people who have CHIP in general are at higher risk of dying, people who have solid tumors who have CHIP are at higher risk of dying. Also some interesting growing literature about looking at how CHIP and different forms of other kinds of clonality might be linked to outcomes with immunotherapy. Some of the earliest work came out of the MSK Cancer Center impact cohort, and this is because of an accident, if you will. So in the MSK assay, they sequence blood as the normal control, as well as the solid tumors. And so work led by Kelly Bolton when she was a fellow at MSK, and Ross Levine and others said, why don't we just flip this on its head and say, let's look at the mutations that are in the blood that aren't in the tumor, rather than focusing on the mutations that are in the tumor that aren't in the blood. And then leveraging the size of this cohort, which is tens of thousands of people at this point, they made some very interesting observations. This is an updated analysis from JCO a year or so, two ago, where they're looking at just if you walk into MSK, and there's all sorts of biases of that because to get to MSK is not the same as a community cohort. But if you did walk in the door and were sequenced on this panel, they saw very different risks of different cancer types across some of the more frequent cancers. So notably, lung cancer and prostate cancer seem to be more common than breast cancer, which had sort of one of the lowest percentages, even after you control for all the things that are associated with CHIP, like age and exposure to toxic prior chemotherapies. Kelly and her group, again, were interested in this lung cancer observation in particular. So they looked at a large-scale population data set called UK Biobank. UK Biobank is about 500,000 people that are, it's hard to describe them. They were basically randomly invited by mail to participate. And, a decent number of people in the UK said yes, so, 500,000 out of the total population. And they looked at people who had, blood drawn and then who developed lung cancer afterwards, and found that, having CHIP was associated with lung cancer, with incident lung cancer, so not just cross-sectionally. And that this seemed to be, particularly, true of certain subtypes like adeno, and seemed to be less true of small cell. But I'll note the numbers get really, 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 really small. And this has been observed by a few other groups and a few other cohorts as well, that CHIP being particularly associated with lung cancer. Our group was intrigued by this, and intrigued in particular by a paper that came out in Nature about a year ago, looking at the environmental exposures of PMI 2.5, and which is a very, very fine form of air pollution, and showing that that air pollution seemed to activate macrophages and be particularly linked to lung cancer in non-smokers. And so CHIP is also strongly linked to inflammatory macrophages. And so one of the things that we were interested in querying is whether these hyperinflammatory CHIP macrophages are also drawn to the lung epithelium by the PM2.5 air pollution, as a mechanism of tumorigenesis. So we also analyzed the same UK Biobank data set that Kelly and her group and others have looked at, and first of all confirmed that patients who whether they smoke or whether they don't smoke, there is a notable increased risk for non-small cell lung cancer amongst individuals who have CHIP. And then we further stratified this by people who have sort of above average PM2.5 air pollution versus below average PM2.5 air pollution, and found a pretty striking association or interaction between the association that even among people who never smoked, those who have CHIP seem to have this sort of enhanced risk if they have increased exposure to air pollution, which fits with some of the mechanistic work that was published a year ago about these inflammatory macrophages. And I think that we're particularly interested in this is that if you look worldwide at where the UK sits and the amount of air pollution they're exposed to, it's, and same with us here in the US, it's relatively benign. There's almost no PM2.5 exposure compared to large parts of the world. And so to the extent that this association between CHIP and non-small cell lung cancer is sort of enhanced by PM2.5, we expect that it'll probably be even greater when we look elsewhere in the world, which is something we're actively working on. And so I think collectively, both on the some of the heart disease and some of the cancer observations, Eric Topol, who I like to think of as a medicine influencer at this point. I used to think of him as a leading cardiologist, but now he's sort of a commentator on Twitter. Put up a blog post a year ago saying that really we're underappreciating CHIP and what CHIP is, and that he thinks we have a marketing problem, that we really should drop the indeterminate potential and come up with something else that should be clonal hematopoiesis of importance potential or something like that. So I told you a bit about what CHIP does in solid tumors, and I want to talk a little bit about our work about CHIP in kidney disease. And this is, I'll focus mostly on some of the epidemiology and do a little bit about mechanism here as well. So, how is CHIP affecting chronic kidney disease? How is it affecting AKI, acute kidney injury? And then what about kidney transplant? Which is all just to say that there's plenty of exciting scientific possibilities of CHIP in the kidney. First, the first paper that was published on this was a few years ago, led by Brian Kestenbaum at University of Washington, looking at CHIP and how that affects kidney function decline in the general population. And so we looked across three very large epidemiology studies: ERIC, atherosclerotic risk and community study, CHS, cardiovascular health study, and MESA, multi-ethnic study of atherosclerosis. And pretty much across all three, individuals who had CHIP were at 25% increased risk of having a 30% decline in their EGFR, which is particularly notable because almost all of these people were very, very healthy at baseline. So, just even with a decade of follow-up, to see this robust association was pretty impressive. We've followed this up now in some unpublished work that has been submitted across a number of other cohorts that have very detailed kidney progression, sort of more in the short term. One is called this CRIC study, and a few others. And again, we very, very nicely replicate this, modest increased risk of CKD progression in people with CHIP. And this, again, is particularly true in people who have non-DNMT3A CHIP. So as I mentioned earlier, about half of people have DNMT3A CHIP across a large number of diseases. DNMT3A does not confer the same sort of inflammatory sort of disease risk as these other genes. We, flipping to AKI, this was published in Nature Medicine a few months ago. At this point, found sort of similar things. So individuals who have CHIP are actually at increased risk of developing acute kidney injury. And this is particularly true, again, in some of the genes that are not DNMT3A, and seems to be at sort of increased risk for patients who have genes that are more uncommon and involved in more of the sort of signaling side. And this is true not just in who gets any kind of AKI, it's also people who have among people who have CHIP, there seems to be a much greater likelihood of having non-resolving AKIs, so the kind of AKI that will then progress to requiring dialysis or other sort of more severe interventions. And we also looked at dialysis in particular, and again, the association with dialysis seemed to be driven by the larger clones. As I mentioned in one of my very first slides, that the more fraction of blood that is taken up by these mutant cells, the worse off you are. So, this is all really exciting. We have great human genetic data, but when you send a paper to review at a top-tier journal, really what they care about is whether this is true in a mouse. And I'm only half joking here. So there's a number of sort of well-established mouse models pioneered by my friend and colleague Sid Jaiswal when he was in Ben Ebert's lab, as well as similar work done by Ross Levine and others, where basically you can just give the mouse CHIP by doing a bone marrow transplant. So you take the mouse, you can irradiate it, that kills off most of the mouse's blood stem cells, or if not all of them, lethally irradiate it, and then you just do a transplant where you put in some CHIP mutated blood stem cells along with wild type cells. And now you have a mouse who has CHIP. There are limitations of this model. There's other ways of doing it without radiation. But this has been sort of used pretty pervasively. And so it's easy to give a mouse an acute kidney injury. You can just basically give it an ischemic injury to the kidney, and you can show that among mice that have the TEP2 CHIP, you can replicate the rise in BUN, rise in serum creatinine, above and beyond the wild type mouse. And so then the question is, why, basically, why are these mice, having worse ischemic kidney injury? And what we found is that the mice tend to have more of these pro-inflammatory macrophages, these CD68 macrophages in the context of TEP2. They tend to be more likely to specifically express NLRP3, which is part of the inflammasome. Inflammasome pathways are sort of very rich history in the CHIP world. And so, these blood cells are going into the kidney, and then when the kidney is injured, they're leading to even worse injury. And as a result, we also then see late effects of kidney injury, including notably worse fibrosis in the kidney in the TEP2 CHIP mice than the wild type mice. And so we put all of that together to suggest, here's the mechanism how a blood cell in circulation, a mutated blood cell, can cause injury in the kidney. And And reviewers said, well, that's really great, but is it really the TET2 mutated cell that's causing this damage? And the neat thing is that we're able to say yes pretty confidently. And that's because when we have the mouse that has CHIP, we do a clever thing that again was developed by Sid and Ben Ebert and others, which is that you just put the mice on slightly different backgrounds, where one mouse is a CD45-1 and the other one is the CD45-2 epitope, and so then you can stain and pull apart that and read it as the chip, the TET2 mutated cells that really are conveying this burden of inflammatory signaling. The last context that I'll bring up is a transplant. People are very interested in the kidney world. What about what happens with kidney transplant? And so patients who get a kidney transplant are at about twice the risk of having CHIP mutation as the general population. We found this across 2 different cohorts, the UK Biobank and all of us. And by and large, we didn't find that they had any greater risk of complications from their kidney transplant or any greater risk of rejection. So again, the significance of this is something that we're still looking into. But yes, patients who have kidney transplants also have more CHIP. So I want to peel back the onion a little bit more and say, now that I've told you that CHIP is in all of these different places, has all of these sort of poor outcomes, how is it that these mutated blood cells are actually acting in a bad way? And sort of how did different CHIP mutations confer differential risks? So this is a project I dreamed up as a postdoc that was like, this is what I'm going to do as part of my early career faculty. And I'm just going to do single cell RNA sequencing and just figure out, within the same person, who has CHIP mutations, which blood cells don't have the CHIP mutations, and I can compare mutated to unmutated. And again, this is not a unique idea, as often happens in science. There's been wonderful work done similar to this by Vijay Sankaran at Boston Children's and Dan Landau, New York Genome Center and Cornell. But it ends up that this is actually really, really difficult. And the reason it's difficult is that these genes that I care so much about, like DNMT3A and TET2, are not actually expressed in peripheral blood at any kind of appreciable levels. So macrophages, by the time the cell is differentiated into a macrophage, whatever effect TET2 or DNMT3A has had on the epigenome, has happened in the stem cell and it has propagated. And so we developed a strategy where what you can use are mitochondrial DNA. Mitochondrial DNA is very dynamic, is constantly acquiring mutations, and is also very easy to enrich from single cell RNA-seq libraries. In fact, often you're trying to get rid of mitochondrial DNA from your single cell RNA-seq. And so we were able to put these things together where we can do DNA sequencing to figure out what the mitochondrial mutation is, and RNA sequencing to then link that mitochondrial mutation to the rest of the gene expression profile. We have a paper that's now out about this, preprint. It's been out for quite a while. And again, Dan Landau, Vijay Sankaran have similar papers if you are excited about doing this kind of thing. And we can use other platforms like the Mission Bio Tapestry platform that can do DNA sequencing but can't really do RNA to confirm that a particular mutated mitochondrial mutation has this property of being linked to the TET2 mutation. And then once we have that, we can then look in single individual's blood and say, what's the difference between the mutant and the wild type cells? And pretty similarly across DNMT3A and TET2, we see evidence of this lineage bias. So even though perhaps 5% or 10% of your cells might be DNMT3A mutated or TET2 mutated, 50% of your CD14 monocytes, or 80% of your CD14 monocytes might carry this mutation. And they might be relatively depleted in T cells. And so this is basically why, or one of the reasons why we think that CHIP, even though it might be a small fraction of your blood in total, confers such a notable disease burden. And then again, I won't go into this, but these mutations, once you are able to identify the single cells that have the mutation, you can start looking at signaling. And if you want to learn more about that, take a look at the paper. I want to spend the last few minutes just talking about management. How are we thinking about patients who have CHIP? Because many of your patients for the oncologists who are part of the Science Academy have CHIP and may require treatment. Again, right now, these are largely incidental findings. Patients who have solid tumors are getting sequenced. But we're also finding that patients are referred to hematology for a blood count abnormality or getting sequenced, and often the blood count abnormality might resolve. It might just be a viral thing, but even though the count abnormality resolves, the mutation persists. So and really wonderful work led by Lachelle Weeks, who's now a junior faculty at Dana-Farber, did her postdoc with Ben Ebert. We helped derive a risk score for clonal hematopoiesis, where you can look at different features of the person and of the molecular profile of the mutation to come up with features that are relatively higher risk or lower risk. And Lachelle and team have developed a great online calculators. You can type this in, have it handy on your phone. But from this work, Lachelle and there have been similar scores derived by others, we're able to find that these mutations or these molecular profiles are not all created equal. That out of the 20% of people in their 70s who have CHIP, there's a very small subset, probably 0.5%, who have sort of the highest risk CHIP. And those individuals are really the ones who should be included in clinical trials, who should be followed more closely. And 90% of people with CHIP are at very, very low risk and have almost no increased progression to malignancy, almost no decreased survival above and beyond people without CHIP, and probably are not the people who are most of interest. And so this is now a way that we can start stratifying risk. This is not the final word on CHIP and risk. There are many other features that we and others are showing can be incorporated into these models. For example, if you have a heterozygous mutation versus a homozygous mutation, that confers very different risk profiles. And then just, again, I know we're running short on time. I want to leave time for questions and discussion, but what are we actually doing for them therapeutically? Right now there are no FDA-approved therapies for CHIP. There are a number of clinical trials underway. There's, I'd say, two different strategies, broadly speaking. One is by targeting specific driver mutations. So these are two trials led by Kelly Bolton, as well as colleagues of hers, former colleagues of hers at Sloan Kettering, where a very small number of people have IDH1 mutated abnormalities, but we do have IDH inhibitors, targeted inhibitors, and so you can use those targeted inhibitors to potentially treat the clone. The other strategy is, I've highlighted it a number of different times now how inflammation is sort of a common final pathway for patients who have several of these different CHIP mutations. And so there are drugs that can be used to treat inflammation. For example, IL-1 beta directed therapies like canakinumab or other inflammasome inhibitors. And these are all trials that are also sort of actively recruiting. And so I suspect that in the next few years, we probably will have more data, at least, to suggest what therapies might be of particular interest. in humans, and this will probably drive further testing for CHIP. I want to thank the many awesome people that have contributed to a lot of the work that I presented today. Our lab dressed up as a Manhattan plot, as a GWAS for Halloween last year, which is a little bit goofy. Wonderful collaborators across the country and around the world, and couldn't do any of this without really significant investment from a number of different sources. So thank you. I'll stop sharing and can
Thanks,
discuss
Alex.
whatever you guys want to talk about.
Yeah, appreciate it. And actually, the Shell Weeks gave us a talk a few weeks ago, so this dovetails nicely with what she presented. I guess I'll open up to the group. Maybe it seems to me as a general statement that more is known about the association of CHIP with various medical issues, let's say, than about mechanism per se. Is that a fair statement?
I think in humans, there's been a lot of these mouse models exist, and then I think the question is always sort of how accurate, how well did these mouse models recapitulate what's happening in people, because we're not irradiating people and giving them bone marrow transplants with CHIP in them more. Well, we do sometimes,
We
but we
are,
try
yeah,
not to.
I hear you.
So,
I hear you.
yeah, no, I think this is sort of the next phase where this is like a genetic disorder that people didn't know existed a decade ago, right? 2014 was 10 years ago. And then to be able to go from that to having some trials in that span is, to me, like utterly mind-blowing. But yeah, no, there's a lot more to do.
Yeah, agreed. Questions from anybody else? Comments?
I kind of missed a little bit of this, but this was great, Alex. The whole connection between the air pollutants. I think with kidney cancer, I feel like a lot of us sort of then chalk up to environmental exposures, particularly because we see a rare number of patients with, let's say, germline inherited risk for kidney cancer development, and certainly some of the other risk factors like obesity and let me see kidney disease or things like that are kind of inherent in the population. So I don't think we have a good sense of what are the real triggers sometimes for a lot of that tumorigenesis. And I feel like a lot of us end up kind of blaming some type of maybe environmental exposure, but the air pollutant thing is interesting. I guess twofold question would be, one, from the air pollutants piece, do we see that in like other parts of the world where there's inherently significant pollution or contaminants that maybe people are exposed to, historically? And then I guess the second would be mechanistically connecting some of that to the AKI piece. Do you think that some of that there's a relationship there in sort of mechanism? Just because I mean, a lot of what you're talking about sort of stems a lot of questions in my mind about patients who have nephrectomy or post nephrectomy and what does renal function look like, and kind of trying to make that mechanistic connection, I guess, to CHIP.
Yeah, no, so I mean, what was, again, mind-boggling to me is that, like, people love saying, oh, it's genes and environment. And then you're like, okay, well, what is the environment? And people are like, well, smoking's part of the environment. I'm like, okay, I can buy that. Like, what else is in the environment? Like, what about non-smokers? And so again, the super cool thing when this paper from Charles Swanton and his group came out is like, it's not all air pollution. It's like a particular, subset of this dust. I'll put it in the chat again. It's just super cool. And then to be able to say like, well, dust of this size, seems to be specifically, activating macrophages and driving them, towards the, particular places in the tumors, and is just, again, and just sometimes you have these insights where you're able to say like, okay, great, everyone knows about genes and environment, but now we actually like have something we can test, and makes it a lot more tractable. And
I think that, like the stories, I guess like from the kidney perspective, it feels like the story is kind of evolving into like this microplastics or what we're ingesting, let's say, that might be depositing within the kidney or, causing some sort of contaminant in that respect. I feel like we're kind of
yeah, no,
at least
exactly.
heading in that direction, which is kind of like way different than the air inhalation piece, right? As another mechanism, but very cool.
But I think, all of these, and I mean, with the kidney, you have so many exposures, right? Because it filters everything. But, how do we take that kind of work to the next level and say, microplastics, you can at least test for in theory. I mean, again, like I'm sure a number of you saw the New England Journal paper about microplastics and
Yeah.
like coronary, atheromas. It's like, whoa.
Yeah, that's right. Yeah, that's kind of stimulating. I
Throwing
feel
out
like a
all my
lot.
Tupperware.
Right.
But I think there's probably, with newer technologies, there's ways of evaluating these things, with mass spec, like what's in the urine, with urine-based metabolomics, urine-based proteomics. So I think that's cool, because then, whether you're a translational scientist or basic scientist or an epi person, then it gives you something very specific to investigate further rather than just environment. Chip AKI relationship, again, for us, it all comes back to the macrophages, which I think are, an important part in your kidney cancers as well. We had a nascent collaboration with a graduate student who was in Tim Rathmell's group, who's now graduated, to look at, what happens when we sequence, take our chip assay and try and look at cells that are in kidney tumors. But I think we did 3 of them, and we found like an SRSF2 mutation in one of them. So like this was with Brad, Brian Field, as you might imagine. And he's now a fellow, and so it sort of moved on, or a resident, I should say, at UCLA. And so, I think there is a lot of opportunity to try and come back to this question about, what are macrophages doing in the tumor microenvironment in the kidney. Kidney cancer happens in older adults, CHIP happens in older adults. I mean, there's a lot of ways that you can line up some of these arrows and think about that. But I think also just in terms of in renal cell, you have this same paradigm of, what is a precancer? How does the precancer transform? Why does the precancer transform? And so with CHIP, from my perspective, it's a great model system to look at that, because you can really easily assay precancers across this huge general population. So now I'm just talking around your question.
No, I mean, it's a, macrophage piece is interesting, obviously, because we see so much infiltration, right? And we think that there's probably some degree of infiltration that also seemingly is enriched in, let's say, higher risk, kidney cancers compared to lower risk. So obviously, from a trajectory standpoint of the history, or the natural history of that disease, we definitely see that difference. It's just been hard for me to link a lot of the CHIP findings outside of just like, I know that we always control for, like, for example, age or, other comorbidities, but it always feels like it's just coming back to like that patient is just like in a different phenotype almost. And I do wonder a little bit, I'm at MSK where I mean, obviously we have a lot of access to a lot of CHIP data, and we've looked at survival curves between, CHIP and no CHIP, but I always wonder a little bit about some of the kind of scary kidney cancers that we see in very younger individuals, and are they biologically have features that are sort of representative of like an older population, like CHIP or things like that, but it's just harder to kind of parse out with the data.
And, it's an MSK, it's a blessing and a curse, for
There's a lot
for people who are sicker, who, come to you guys after they've already failed therapy across all of the, all of northern New Jersey, and then they've ended up in New York City. So as someone who grew up in New Jersey,
that's a good way to put it. Yeah. Thanks though.
I'd love to ask a quick question, Alex, if since I can actually pick your brain while you're here. So last year I did like this analysis in the UK Biobank looking at cancers that have a higher risk of cardiovascular predisposition. And so what I was interested was looking at markers that could maybe identify this higher risk of cardiovascular related events. And so I looked at the impact of carriers of mosaic chromosome alterations in patients with these different cancers, and kidney cancer was one of them, and it was highly associated with cardiovascular related mortality and coronary heart disease related mortality. And so at the time, I didn't have access to the whole exome sequencing data from UK Biobank. Like I had only a half of the version of it. So I wanted to look at the whole cohort, and recently we did that. We were able to do it on the whole cohort. And what we found was that patients with CHIP mutations with kidney cancer actually don't have that exacerbated risk, higher risk of CVD. But that really, when you combine the CHIP and MCA, it's really the MCAs that are driving that impact. So I was wondering, I was thinking, well, so it seems to be very a distinct sort of relation. It's like they don't sort of overlap each other. So I thought, like maybe that's what it is. So I'm wondering, what are your thoughts or insights on that?
Yeah, it's interesting. I think one of the challenges, I don't know off the top of my head how many renal cell cases are in UK Biobank, but it's not that many is my guess.
Yeah, it's 1900.
Yeah, so I mean, it's not nothing, but
yeah, it wasn't. Yeah, it's not superb.
Yeah, but the challenge is like if you're starting with 2,000, or broadly speaking, if you're starting with 2,000 people, and then 5% of them, let's say, have the thing that you're interested in, and maybe 5% have this other thing, then sometimes there can be a lot of just somewhat random fluctuation just because the numbers are small. And then, so I think the key would be if you're able to replicate that somewhere else, in all of us, would be something that is probably more worth speculating about or hand waving over. But yeah, so I think in general, MCAs haven't had particularly notable cardiac phenotype other than like the basically 9P, which is JAK2. But I think there's still lots to learn for sure. And then yeah, to the point made earlier, like in what way is it a driver, and which way is it sort of a stratifying? So I think interesting question.
Thank you.
Thanks, Zach. We're right up on time, so appreciate your time and expertise today, sharing with the group. It was great.
Thank you.
All right, thanks all. See you soon.
Thank you.
All right.